Welcome back, everybody. We are now going to move on to the next lesson, lesson 3 in our communicable diseases. And this lesson is going to be on the structure of our antibodies, and you can find the information that you need for this on page 320. I've also included some sheets which you can use to help you to take some notes as you go along, and they include some of the diagrams that you'll find on this presentation. So lesson 3 is going to be about the general structure and function of antibodies, and we're also going to be looking at how they behave in order to destroy the pathogens that get into the system. So this is looking at antibodies in relation to the specific immune response. So before we do the specific immune response, I wanted to make sure that we had a good understanding of the antibody structure, and I hope you appreciate my little joke there on the right. If we look at this slide, what we can see is we have two molecular models for antibodies. Most of you will remember that you have done a PAG, which is molecular modelling on the computers in the IT suite. And I think we particularly looked at things like collagen and rubisco and sometimes hemoglobin, that type of thing. So what we can see here is we can see two different versions of antibodies depending upon the style of the molecular model that we're using. So I think it's sort of like left hand side one looks like stick and ball one, and the one on the right looks like it's more of a sort of a ribbon and backbone type of structure. But what I wanted you to think about with this is that in some of the questions they obviously like to use the sort of PAG activities as a question, so you could be given a picture like this and be asked to describe what you can see or identify various different parts of the molecule. So just remember that that is something that could be applied to something like the structure of antibodies. So we're going to have a look at antibody structure, and on the right hand side there I've got a generalized diagram showing an antibody. Now quite often questions will ask you things like describe the structure of an antibody, or perhaps another kind of question where they might ask you to compare the structure of an antibody to something like hemoglobin or collagen. So in that instance what we have to do is we always have to remember that antibodies, also known as immunoglobulins, are basically proteins, and because they're proteins we always start from the very beginning and make sure that our description of an antibody begins with all the fundamental knowledge that we should be recovering at this point. So this was also a little bit of a revision really of your protein structure. So the first thing to state, if asked anything about antibodies, is that they are proteins, and therefore they're going to be made of amino acids. And of course the primary structure of our protein is coded for in the DNA, so we have genes which are there specifically to code for antibodies. The amino acids are joined by peptide bonds, and the proteins themselves belong to the group of globular proteins, and the name itself is an indication of that, because antibodies are also known as immunoglobulins, and that tells you that they are globular proteins, which therefore means that you already know something about them. You'll know that they are biologically active, they're also soluble. They'll also probably have quite a lot of alpha helix in their secondary structure. So we can refer then to our primary, secondary, tertiary and quaternary structure, and very often it's a good idea as well to refer to the types of bonds that will stabilise each of these different levels of protein structure. So we're probably going to say we can refer to the fact that the primary structure is coded for in a gene. The secondary structure probably will have a lot of alpha helix, and that's stabilised by hydrogen bonds. We will have a tertiary structure. The tertiary structure involves hydrogen bonds, hydrophobic hydrophilic interactions, disulfide bonds, and if you just look at the right there you can see that we've actually got disulfide bonds labelled. So we know that they are playing a part in the structure here. We've also got a quaternary structure, because if you look at the diagram you can see that we've actually got a molecule that is made of more than one chain. In fact there are 4 chains in an antibody, 2 smaller chains which are called the light chains, and 2 longer chains which are called the heavy chains. So the proteins then, these antibodies are very large and molecules, and they're also soluble, and they will circulate in the blood plasma. So this is the diagram that you have already got, and you've got this blank diagram. So what I suggest you do is that while you're listening to this, you take this opportunity to annotate this diagram, and I'm going to be going through the annotations that I've got on this presentation, and these are the bits of information that you're going to need to know. So the first point is that we're going to label up the longer of the protein chains, and this is your heavy chain here, and we've got 2 of them. Now you'll notice that in the middle of this heavy chain we have this flexible region, and it's called the hinge, and this is where some of the amino acids are a little bit more spread out. They're not so tightly bound to each other, and this allows an area in the antibody which has a little bit of flexibility of movement, and that actually assists the antibody in attaching to the pathogen. The 2 shorter chains are just called the light chains, and they're at this top end of this Y-shaped structure. In yellow we have got disulfide bridges. Now disulfide bridges form between the amino acids cysteine, and they're very strong covalent bonds, very difficult to break. So as I've mentioned to quite a few of my groups, when you have molecules which are recovered from the sulfur bacteria, they live in very hot conditions, and they can only survive there because their proteins have a lot of these disulfide bridges, which mean that they are very tolerant to high temperature. And we'll come across that again a little bit later in year 13. So the disulfide bridges are holding the heavy and the light chains together, so they're in fact stabilizing the quaternary structure for this molecule. Down in the lower section of the heavy chain we have this region in blue called the constant region, and this is an area of the antibody which doesn't change. It's the same for every antibody. Every antibody will have this same sequence of amino acids, and that's really important because actually that constant region is the part of the antibody that phagocytes will always recognise, and they recognise them on the basis of their shape, because that's basically how all molecules operate and how all processes occur inside cells. So it can only work because one molecule is responding to another molecule. So if we look at the other end of this molecule, in red we have the variable regions. So this is what distinguishes one antibody from another. So in this variable region we have a sequence of different amino acids, and those different amino acids can vary, and it's about 110 different amino acids possible, and this creates the huge different variety of antibodies that you would get. So the sequence of amino acids in this variable region will subtly alter the shape of that variable region, and that therefore allows the different antibodies to attach to the various different antigens that you would find on pathogens. So the specificity of the antibody depends upon this variable region. So when an antigen is located, it will be able to bind to this antibody, or should say the antibody is able to bind to this antigen, and it forms the antibody-antigen complex, very much in the same way as you get with receptors and things like enzymes binding to their substrate. So very similar sort of thing. They have to be complementary, and that's what we always talk about. We talk about this complementary binding between the antibody and the antigen. And we've got 2 of these variable regions on an antibody. So in fact, one antibody could bind to 2 different pathogens, and that helps to clump together some of these pathogens, which can obviously really help in the fight against the spread of this pathogen inside an organism. So this brings us on to antibody action and agglutination. Now antibody action is, there's quite a lot of different ways in which the antibodies work, and the first one we're talking about here is agglutination. I've just mentioned it on the last slide. So agglutination is basically where the antibodies start to attach themselves to the pathogens, and as we've mentioned before, because we've got 2 different attachment sites on the antibody, they can in fact attach to more than one pathogen, and that causes them to clump together. And when they clump together, that means that they can't spread through the organism as well as they would have done, because they're now much larger than they were. And also the non-variable region is signalling to the phagocytes, and the phagocytes can come and engulf them, and engulf a whole group of them in one go. So that speeds up this whole process of destroying the pathogens. So the next type of antibody action is neutralisation. One of the things that a lot of pathogens do is that they release toxins as a byproduct of their activities, and these toxins can make you really unwell. So the actual presence of these proteins, usually, that they're releasing can interfere with various activities in your body. So, for example, some toxins, what we call neurotoxins, what they do is the toxin that's released gets into the synapses and prevents the normal transmission of electrical signals at various different parts in the brain or other parts of the body, and can lead to things like paralysis and so on. And so getting rid of some of these toxins can be just as important as actually getting rid of the microbe itself. Obviously, getting rid of the microbe will also mean we won't have any more of the toxins. But if you can just actually act on the toxins, then the pathogen becomes less dangerous. So in this instance, we've got this pathogen releasing these toxins, obviously much bigger than the pathogen really, but and we've got antibodies, and these are referred to as antitoxins. So they can stick to the surface of these toxins, and therefore these toxins are now not going to be able to do what they were going to do. So for example, this toxin might have been going to bind to a receptor in a synapse. Well, because it's got antibodies all over it, now it can't bind, so it's going to make it much more difficult for it to actually cause any problems. The next thing is opsonization, which we've already mentioned in a previous presentation. So opsonization, as you know, is this process by which the antibodies, and certain ones like immunoglobulin G, for example, will bind to the pathogen. And when they do that, that is a signal to the phagocyte, which we've mentioned with the first one, the first antibody action, and the phagocyte is able to identify that these antibodies are now attached to these pathogens. So that will signal, and the phagocyte will come along and ingest them and digest them. Now, opsonization can also be because when the antibodies bind to the walls, they can actually interfere with the surface of the pathogens, so the cell wall, and it makes it more leaky, so they eventually absorb so much water from the tissues that they explode, which is obviously in fact how quite a lot of antibiotics work. Antibiotics often work by causing the bacteria or the pathogen to become more porous, so that they absorb so much water that they can explode. The next thing that antibodies can do is that they can block receptors. Now, a lot of viruses find their way into your cells by landing on your cell's receptors, so your glycoproteins, and using those glycoproteins and other proteins as a means of getting into the cell. What a virus normally does is it lands on the receptors and then inserts its DNA or RNA into the cell, and the DNA or RNA then starts to give the cell instructions basically to make viruses, and the cell will start to use its own resources in order to create viruses, to the point where eventually the cell is destroyed and then all the viruses break out and infect nearby cells. So if you can actually prevent those viruses from entering those cells, then you have made those viruses totally ineffective. They won't be able to carry out any more damage in your cell. So when we get a viral infection, one of the things that the body does is it prevents the viruses getting in, and that's how a lot of antibodies work with viruses. So normally, as you can see here, we've got the virus sitting on the surface, just about to inject its DNA or RNA into the cell, and the antibodies there on the left have stuck to the virus now, and that means that those receptors that are on the surface of that virus are now useless and cannot get the virus into the cell anymore. So that's quite an important That's quite an important role of the antibodies. So which cells are going to produce antibodies for us? Now you can see a picture here on the right, and it's a photograph showing us a lymphocyte. Now lymphocytes are one of the cells that produces antibodies. Now antibodies can sometimes be stuck on the surface of the cell, and sometimes they can be released into the plasma. So we're more interested in the ones that are released in the plasma in this instance, but the ones on the surface of the cells do a similar job. So lymphocytes are noticeable by the fact that they have a very large nucleus, and that nucleus plays a very big part in the manufacture of these proteins. And if we look at the cell ultrastructure, what we'll find is because they're making lots and lots of antibodies, they'll have a very highly developed rough endoplasmic reticulum, lots of Golgi, and you'll need quite a lot of cytoskeleton in order to move everything around between the RER and the Golgi, and to carry the vesicles carrying the antibodies out of the cell. So the ultrastructure refers to all the organelles inside the cell, and you could be asked a bit about that and asked to explain how a lymphocyte will be modified and adapted to be able to do its job better. So a little test or a little quiz for you now. I've got some questions for you to have a quick look at. So all you have to say is whether they're true or false. So I'm just going to let you have a look at these different questions. Okay, so you've had a chance to have a look at them, and I'm just going to go through the... So let's have a look at the answers then. The first one, antibodies have a variable region specific to pathogens, antigens. Well, that's going to be true. Antibodies contain a sulfide bridge. Well, nearly, but it's actually a disulfide bridge, so that would be false. An antigen-antibody complex is formed when an antibody binds to a pathogen's antigens. That's true. Agglutination makes it easy for pathogens to overwhelm the immune response. Well, that's false. Agglutination actually makes it easier for the immune response to deal with the pathogen. Neutralisation only occurs with viruses. Not necessarily. Bacteria can release toxins, and they can be neutralised by the antibodies. Optimisation causes phagocytes to swell and burst. No, it does the opposite. It actually causes the pathogens to swell and burst. Antibodies can stop viruses entering host cells. Well, that's true. And antibodies remain in the blood plasma indefinitely. That's false. Antibodies only remain in the blood plasma for as long as they're required. They have a relatively short lifespan, and eventually they will disappear. So it is possible to identify whether somebody has had a disease by whether or not they've got antibodies, but maybe not over long periods of time. If somebody has become immune to something, it's not necessarily because they've permanently got antibodies. It's because they've actually got memory cells which have the ability to make antibodies whenever they're required. I hope you found that useful, and that you've managed to find the resources to help you make notes as you went along. The next PowerPoint is going to be on the specific immune response, so we'll be looking at how these antibodies are being created and how they are responding to the pathogens that are present. Thank you very much, everybody.